Right, well, welcome to chapter number 17. Here we're going to look at how the information encoded in your genes is decoded and used to make proteins, literally the flesh and blood of your body. Alright, so the information in genes then is the nucleotide sequence within the gene. That's the sequence of the A's, T's, C's and G's. That is the genetic code of the gene. So think about an analogy. Take any word like the word genetic. The sequence of letters gives that word its meaning. That tells us what that word is and what it's mean. So in a very similar way, it's the sequence of nucleotides in a gene that give the gene its meaning. So genes, remember, are very long stretches of DNA, maybe 1,000 or 2,000 base pairs long, and I'm going to make up a genetic code right now, just a very short one though. And I'm going to stop there. That could go on for 1,000 or 2,000, and of course that's one strand of the DNA molecule. I'm going to draw the second strand, and you know the DNA binding rules. And of course you know that this makes a base pair, and there can be again 1,000 to 2,000 or even more in many genes. And a gene is simply a length of DNA that codes for a polypeptide. So that gene sequence is interpreted and it directs the synthesis of a protein. Now proteins literally form your flesh and blood. And if you remember, proteins have a huge variety of roles. You can have structural proteins like collagen that forms a lot of the structure of your skin. You can have enzymes like salivary amylase, which facilitates a chemical reaction. You can have signaling proteins like insulin, or oxygen carrying proteins like hemoglobin. So proteins have a huge variety of roles in your body. So the proteins then, directly or indirectly, affect your phenotypes and your biochemistry, and it's really what makes you work. All right, so let's have a look at this picture. Here we've got a albino deer, and I believe this picture was taken of a herd in Germany, and it created quite a lot of controversy when this deer was first spotted. But this deer is an albino because it inherited two defective copies of a gene, and that gene, the functional copies of the gene, would code for an enzyme which are in the biosynthetic pathway to produce the pigment melanin. So the pigment melanin is what makes those normal colored deer their dark brown color. So if you remember, a biochemical pathway kind of works like this. You've got substance A gets converted to substance B, and substance B gets converted to C, and then substance C gets converted to D. Now let's just say that D is melanin, the brown pigment, and A is some precursor chemical. And in order to convert A into B, you need an enzyme. Let's just call it enzyme 1. And then in order to convert substance B into C, you need enzyme 2. And to go from C to D, you need enzyme 3. Now remember, the enzymes are proteins and they're produced as a result of gene expression. There's a gene that codes for that protein. So in the case of the albino deer, it doesn't have any melanin. It can't produce melanin because one of the enzymes in the biochemical pathway is non-functional. It doesn't work. Both copies of the gene that code for that protein don't work. So let's just say, for the sake of argument, it's this enzyme number 2 that doesn't work. It means that the gene that codes for that enzyme is non-functional in this case. So if enzyme 2 doesn't work, we can't get substance C and we can't get melanin. In other words, everything sort of stockpiles at B. Okay. And albinoism isn't just restricted to deer, of course. Here's a group of pretty startled looking raccoons, and there you've got an albino raccoon which has inherited 2 defective copies of one of the genes which is responsible for the production of an enzyme in the melanin production pathway. Alright, so I want to introduce this term gene expression then. That's the process where DNA, the gene itself, directs protein synthesis. So when I say gene expression, I mean it's where a gene directs the synthesis of a particular protein. And that's the process we're going to look at more closely in this chapter. And that occurs in 3 stages. All right. So you've got stage number 1, which is a stage we call transcription. And I'll briefly describe it now, but we'll look at this in much more detail. So transcription is where we take the gene, the genetic code, and we're able to produce an actual molecule from that code. We're able to produce something called pre-messenger RNA. The next part is RNA processing. It's where we take our pre-messenger RNA and we process it to produce functional messenger RNA. And the last part is called translation. That's where we take the information carried in RNA and we use that to direct the production of a protein or a polypeptide. So gene expression literally takes the code, the information in the DNA, and it interprets that information and it produces actual molecules, proteins. And so we're going to look at each of those 3 stages in more detail. Alright, so first we're going to go back in time a little bit and we're going to see how the link between genes and proteins was discovered. So we're going to go back over 100 years ago, 1902, and of course it was a British physician, Archibald Garrod. He was the first person to suggest that genes dictate phenotypes through enzymes that catalyze specific chemical reactions. Now at the time they didn't even know what genes were made of, but this was something that he proposed. And let me tell you why he came up with that idea. So he was aware of this rare inherited genetic disease called alkaptonuria. It's often called black urine. And you can see by this picture on the left that urine is usually that color. But if you have this rare inherited genetic disorder, you produce a substance called alkaptone. And that substance is released in your urine, and on exposure to air it oxidizes and forms this black color. That's why it's called black urine. So he was kind of fascinated, and he thought that symptoms of that inherited disease and all other inherited diseases were put because of your ability to synthesize specific enzymes. So he said you can't make certain enzymes, so you've inherited a defective gene, which means you can't make a specific enzyme, and if that enzyme is non-functional, then that's going to cause some malady in the body, some kind of disease that you're going to inherit. So that was very impressive, honestly, for over 100 years ago, but it wasn't quite the whole story. He was very, very close, but not quite the whole story. Alright, so George Beadle and Edward Tatum wanted to further explore that idea, and that is the idea that genes produce enzymes, and through the action of those enzymes that affects your phenotype. So they wanted to further explore that idea. And they worked with a bread mold called Neurospora crassa. And here you can see we've got some moldy bread. So some of the bread that you have going moldy in the back of your fridge, if it's got that coloured mold on, then it might be Neurospora crassa. It's quite a common mold, and you can see on the image on the right, that's what it looks like when it grows on agar in a petri dish. Now it's an interesting fungus. It's actually haploid, which means it's only got a single copy of every gene. Remember, you are diploid. You've got homologous chromosomes, and you've got 2 copies of every gene, 1 copy on each chromosome. Neurospora is different. It's haploid, only 1 copy of every chromosome, and therefore a single copy of every gene. That makes it much easier to work with, because any change in the gene will have a direct effect on the phenotype. You don't have to affect both genes in order for the phenotype to be affected. Okay, so Beadle and Tatum then exposed the bread mold to X-rays. And when you expose an organism to X-rays, you are going to damage the DNA, creating mutants. Now, they specifically wanted to generate mutants that were nutritional mutants. So the mutation would have some impact on the ability of the Neurospora mold to grow on certain media, to utilize certain food sources. But when you expose this fungus to the X-rays, it's going to blitz and mutate many different genes. They just wanted to home in and focus on the nutritional mutants. Alright, so I've got to explain a little bit what this minimal media means. So normal Neurospora crassa, ones that's not been mutated, one that grows on your bread in the fridge, that is able to grow with very modest nutritional requirements. In fact, you can give it some inorganic salts, some glucose, and the vitamin biotin, and using its armory of biochemistry and all of its enzymes, if all of its genes work well, it can make everything that it needs to grow and survive. In particular, it can use those minimal nutrients in that minimal media, which again is just inorganic salts and glucose and the vitamin biotin, to produce all 20 of the amino acids that it needs to grow. All right, that's quite remarkable. So if you were to expose them to X-rays and cause mutation in some of its genes, that means that the enzyme products from those genes might not work properly, and so some of its biochemical pathways might not complete. And since the amino acids are produced at the end of different biochemical pathways, if some of the enzymes in that biochemical pathway don't work, then it won't be able to produce the amino acid, and it will be unable to grow. Alright, so let's have a look at this diagram then. So over here we've got the fungus, and you can see there was a little plug that was put at the center, and that plug would have the fungus on it, the Neurospora crassa, and the agar, the media in the petri dish, has a minimal media, alright, in both of these plates. A very minimal media, and these are 2 wild type strains of the fungus, and they're able to produce all of the enzymes needed to make all of the amino acids from that minimal media. Over here, on the other hand, we've got a mutant. And so on that little plug, a strain of the fungus was placed, and there's some deficiency somewhere. Some of the genes don't work, so some of the enzymes don't work, and it's unable to make some key amino acids. So that's what Beadle and Tatum wanted to find. So the X-rays then cause mutations in some of the genes, and that would make either the genes completely inactive, or it might mean that the genes might code for a defective protein which doesn't work properly. And Beadle and Tatum, together with some of their co-workers, they narrowed down, of all the mutants and strains of mutants that they created, they narrowed down to 3 classes of arginine deficient mutants. Now this substance arginine, that's an amino acid, and it's a critical amino acid that the fungus needs in order to grow. So they generated these 3 classes of arginine deficient mutants. So that's 3 classes, 3 strains of the fungus that were unable to produce the amino acid arginine, and therefore unable to grow on minimal media unless it was supplemented with arginine. Alright. And the 3 classes of arginine deficient mutants, it turns out each of those 3 classes lacked a different enzyme that was essential for synthesizing arginine. So based on their experiments, and I'll elaborate on their experiments in a moment, they developed a 1 gene, 1 enzyme hypothesis. And that states that each gene dictates or directs the production of a specific enzyme. So that was their idea or their model at the time based on their results. So let's have a little closer look at the experiments that they. So here you've got some precursor substance which is found in the minimal media. So if you're a Neurospora crassa fungus, you need to produce enzyme A, and that will convert this substance, this precursor, to this substance ornithine. Now remember, this is a biochemical pathway. Now ornithine will be a substance that this enzyme B will then convert into citrulline. Okay, and then we need enzyme C to convert citrulline into that key amino acid arginine. Now remember, there's a gene which is responsible for producing enzyme A, and there's another gene that's responsible for producing enzyme B, and another gene that's responsible for producing enzyme C. So when you expose the mold to the X-rays, if it knocked out that gene, you're not going to produce that enzyme, you're not going to produce ornithine, and so you cannot produce arginine, and you're unable to grow on the minimal media. So that would have been one class of their arginine deficient mutants. So let's just say that the X-rays left this enzyme, the gene for enzyme A, intact, but it caused this gene to be defective. Well, then you're not going to make enzyme B. You can still make ornithine, but you can't convert ornithine into citrulline, and so you're unable to make arginine, and again unable to grow. So that would be their second class of mutant. Their third class would be a mutant where the gene responsible for making enzyme C was bad. So they could still make enzyme A, they could still make enzyme B, but enzyme C doesn't work. And so they're still unable to make arginine and unable to grow. So they're their three classes of mutant. Alright, so here's actually how they did it then. They had their wild type cells. These are the normal Neurospora crassa mold cells, and they would grow them on this minimal media, and they should be able to grow on the minimal media just fine because they're the wild type and there's no mutation. And then of course they got their fungus, they're exposed to the X-rays, and that made lots of mutant cells. And so anytime the fungus was unable to grow and divide and look just good on the minimal media, they figured that must be a nutritional mutant. All right, so then they would take those nutritional mutants and they would go through some further screening that I'll show you in a moment, and they would specifically identify the arginine deficient mutant. All right, so let's dissect this table then. So the wild type, of course, can grow just fine on the minimal media. And if we were to give the wild type ornithine, it will grow just fine. And if we give it citrulline, it will grow just fine. And if we give it arginine, it will grow just fine. So it can grow on all of these different types of media. Our class 1 mutants, however, we identified, or they identified those by giving them the minimal media, and of course they were unable to grow. But then they gave them media supplemented with ornithine and citrulline and arginine. And if they could grow on the minimal media with ornithine, citrulline and arginine, then that was their class 1 mutant. So their class 2 mutants, they identified as follows. They would see if they could grow on the minimal media, and if they couldn't, then they would see if they could grow on the minimal media with ornithine. And if they couldn't, but could grow on minimal media with citrulline and arginine, then they identified those as their class 2 mutants. And similarly, their class 3 mutants, not able to grow on minimal media, not able to grow on the ornithine supplemented minimal media, not able to grow on citrulline supplemented minimal media, but they could grow on the minimal media if it had arginine. So that's how they produce their class 1, class 2 and class 3 mutants, by doing that experiment. Alright, so this table helps explain a little bit more about the biochemistry and what mutations occurred in what genes to generate those different media. So here we've got our wild type. And the wild type, of course, has a fully functional gene A, so it can produce enzyme A and convert the precursors into ornithine. Fully functional enzyme B, gene B, sorry, can make enzyme B, convert ornithine into citrulline, and it's got a fully functional gene C, so it can make enzyme C, convert citrulline into arginine, and our wild type can grow just fine on that minimal media. Now, the class 1 mutant, on the other hand, it's got a mutation in its gene A, so it's unable to produce enzyme A. So it can't convert the precursor into ornithine. But of course, if we provide it with ornithine, then it's got enzyme B and enzyme C to complete the metabolic pathway, and it can grow. Mutant class 2, it was unable to grow on the media that was given the minimal media and ornithine. Now the reason for that is it's got a defective gene B, so it can't produce enzyme B, so it's unable to convert ornithine into citrulline, so it can't grow. But if we give it citrulline, because it's got enzyme C, then it's able to grow. Similarly with our class 3 mutant. So there was a mutation in gene C, it can't produce enzyme C, so even if we give it ornithine and citrulline, it still can't grow because it can't make arginine. But if we give it arginine, then it's able to grow. Alright. Alright, so based on these results, Beadle and Tatum developed a one gene, one enzyme hypothesis. And that stated that each gene dictates or directs the production of a specific enzyme. Now that is really good Nobel Prize winning stuff, and of course Beadle and Tatum were they shared a Nobel Prize for this back in I think it was the 1940s. I can't remember when they got the Nobel Prize, but this was Nobel Prize winning stuff. Now many proteins aren't enzymes, so the hypothesis was later revised to say that one gene makes one protein. But hold on, not so fast. Many proteins are composed of several polypeptides. Again, cast your mind back to chapter 5 and we talked about the quaternary structure of proteins, where there's more than one polypeptide chain which comes together to form a quaternary structured protein. So, many proteins are made of several polypeptides, but each polypeptide has its own gene, or the synthesis of that polypeptide is directed by its own gene. So Beadle and Tatum's hypothesis is now restated as the one gene, one polypeptide hypothesis. The information in a gene codes for one polypeptide. But it's common to refer to gene products as proteins rather than as polypeptides. Okay, so let's have a look at some of the molecular players in the journey from gene to protein. One very important group of molecules is RNA, and that stands for ribonucleic acid. So it is a nucleic acid, which means it's made of nucleotide monomers, and it's a very close relative to DNA, but with several key differences. So let's have a look at those differences. The first one is that RNA is single-stranded, which means a strand of RNA only has two ends. Now the molecule can be very, very long and it can be folded in very complex fashion, but it's different to DNA. DNA is double-stranded and so therefore has four ends. So the next key difference is that it contains the sugar ribose sugar, whereas DNA contains the sugar deoxyribose sugar. And the third key difference is the nitrogen base that RNA contains. It contains the nitrogen base uracil, not the nitrogen base thymine, which is found in DNA. So we use the letter U to indicate uracil, and it's the complement to adenine, A. So uracil's in RNA, not found in DNA. It replaces thymine in some ways. We use the letter U and its complement is A, adenine. Alright, so I want to draw a diagram to illustrate those key differences between RNA and its close relative DNA. So the first thing I'm going to do is draw a uracil nucleotide. So there's the uracil nitrogen base, which is covalently bonded to ribose sugar. And of course the sugar is then bonded covalently to a phosphate. So that is a uracil nucleotide. Now I'm going to draw an adenine nucleotide. There's my A, covalently bonded to the ribose sugar, and there's my phosphate. Now to join these two nucleotides together, we covalently link the phosphate to the sugar. So I'm just going to draw a couple more nucleotides to carry on with this strand. I'm going to add, I wouldn't add a thymine, remember, there is no thymine, so I'm going to add a, if I can get my pen to work, a guanine. So there we go. There we've got a guanine nucleotide. Let's join those together. And I'm going to add, just for kicks and giggles, another guanine. And then I'm going to add a cytosine. And I could keep going. All right, it just depends how long the RNA molecule is. And they can be hundreds or thousands of nucleotides long. Now just remember, it's a single-stranded molecule, so it's got 1, 2 ends to it. Okay? All right, good. The other differences are that it contains uracil, not thymine, and it contains ribose sugar, not deoxyribose sugar. So they're the three key differences. I recommend that you draw this. Draw it and label some things, and that will help you remember it. Okay, so there are three different kinds of RNA that I'm going to talk about, and I want to go over the structure and role of each. So the first kind of RNA I'm going to talk about is this one. mRNA is what we abbreviate it to, but the M in mRNA stands for messenger RNA. Now, messenger RNA is transcribed from DNA in the nucleus, and I'll talk about the process of transcription in more detail later. But we derive messenger RNA from DNA, and that happens in the nucleus because that's where the DNA of the cell is found. Now, once the messenger RNA molecule is transcribed, it gets processed or modified, and then it moves out of the nucleus. It literally snakes out of the nucleus through a nuclear pore, and then it attaches to a ribosome, which is found in the cytosol of the cell. Sometimes the ribosomes are bound to the ER, and sometimes they're free, but in close proximity to the ER. Now, within an RNA molecule, there are codons. And this is a very important word that I want you to remember and understand. So a codon is a group of 3 nitrogen bases. And I'll show you how they interact with something called an anticodon later. But for right now, I just want you to understand what codons are and the nature of the messenger RNA molecule. And to do that, I'm going to draw a very short strand of messenger RNA, and I'm going to include the codons. So let's draw a strand of messenger RNA. And I'm going to draw a fairly short one. So I'm going to say there's a U and an A and a G. So there are the nitrogen bases. Now I'm not going to draw the sugars and the phosphates in the backbone just for time. So I'm going to represent it like that. So then I'm going to draw a little bit more of the messenger RNA molecule. I'm going to say we've got a C, and an A, and then maybe I'll do a U, an A, and an A, and I'll again draw the backbone, but I won't bother putting in the sugars and the phosphates. Alright, so there we've got a very short section of messenger RNA, and it could go on to be literally hundreds or thousands of nucleotides long. So a codon then is simply a group of 3 nucleotides. This would be one codon. Okay, and there's another one right next to it. There's another codon. And there's another codon. So in that short section I drew, there are just 3 codons. So a codon then is simply a group of 3 nitrogen bases. Alright, so now let's look at the second molecular player, and that's tRNA. And the tRNA stands for transfer RNA. And it's also transcribed from DNA in the nucleus, just like messenger RNA. And once it's transcribed, it moves out of the nucleus through the nuclear pores into the cytosol. Now, the transfer RNA has a part of the molecule we call an anticodon. And the transfer RNA also carries an amino acid. And that amino acid is very specific to the anticodon. And it picks up its amino acid after it leaves the nucleus in the cytosol. Alright, so here are some diagrams from your book of transfer RNAs. So this one here is a two-dimensional structure representation of transfer RNA. Now, as you can see, it's got a 3' end and it's got a 5' end, indicating there's only 2 ends. That shows you it's single-stranded. And you can see how it's folded. Those dotted lines in there indicate that those are hydrogen bonds joining those nucleotides together. So here's this transfer RNA's anticodon. The anticodon is simply a group of 3 nucleotides at this location on the transfer RNA molecule. And in this case, the anticodon is an AAG. Alright, now up here, on this part of the transfer RNA, that's where the amino acid is attached. Alright, so we can also draw a transfer RNA molecule as a 3-dimensional structure, and you get a better idea of how the folding is in 3-dimensional space. We've still got our anticodon down there, and we've still got the place where the amino acid is attached. Now, just to keep it simple, your book uses this symbol to indicate a transfer RNA. And there, of course, we've got our anticodon. In this case, we're showing directionality 3' to 5' end. And this is the amino acid attachment site, the place where a single amino acid would be attached to this one transfer RNA molecule. Alright, so there's just another diagram of the two-dimensional representation of the transfer RNA. Again, the important parts are that it's single-stranded. There's one end, our 3', there's our 5' end, and you can see that the molecule is quite folded. We've got hydrogen bonds holding together. There's our, oops, pen's going a bit crazy. There's our anticodon down there, an AAG, and it's up here where our amino acid would be attached. Okay, so why don't you draw this along with me, again, just to help refresh you. Okay, so we're going to draw a transfer RNA, single-stranded. We're going to draw it in 2 dimensions first, and it kind of looks a bit like this cross. Okay, so down here is the anticodon, and there would be 3 nucleotides down there. I'm going to draw a different anticodon. I'm going to put a U and a C and a G as this anticodon. And up here is where the amino acid would attach. Now again, your book uses a symbol to represent the transfer RNA, and it kind of looks a little bit like this. We've got our anticodon down there, and I'm going to draw the U, C, and the G anticodon, and then this is where your amino acid, and I'm just using alpha alpha as shorthand for amino acid. That's where your amino acid would attach. Okay, so that's our transfer RNA, our second player in this game. Okay, so let's have a look then at our third player in this game. We've got rRNA, and the R in rRNA stands for ribosomal RNA, and it's ribosomal RNA which is largely what forms ribosomes. So ribosomal RNA is also transcribed from DNA, but is transcribed in the nucleolus. Now the nucleolus, if you remember, is a dark region within the nucleus of eukaryotic cells. It's not membrane enclosed, it's just a darker region within the nucleus, and it's the location where ribosomal RNA is transcribed from DNA. Now, once it's transcribed, RNA also moves out of the nucleus. It moves out through the nuclear pores, and it goes into the cytosol to the endoplasmic reticulum. And we have bound and free ribosomes. And if you remember, the free ribosomes are in the vicinity of the ER, the endoplasmic reticulum, and the bound ones are actually embedded in the membrane of the endoplasmic reticulum. So ribosomes then are the site of protein synthesis. It's where amino acids are joined together to form a protein. Now, ribosomes themselves consist of 2 subunits. In eukaryotic cells, we've got a small ribosomal subunit that has a size of 40S, and then we've got a larger ribosomal subunit that has a size of 60S. Now the 40S and the 60S refer to sedimentation coefficients, and it's a measure of how readily these subunits precipitate out in a centrifuge. Now a complete ribosome is composed of a small subunit and the large subunit, and when they're joined together they form a complete ribosome which actually has a sedimentation coefficient of 80S. So eukaryotic ribosomes have a sedimentation coefficient of 80S, and they're known as 80S ribosomes. Now I know it seems kind of odd that the small ones have a coefficient of 40 and the large ones of 60, and if you add these up it makes 100, but when they're joined together they only have a sedimentation coefficient of 80. So let's have a look then at the structure of a ribosome. So this diagram's from your book, and let's get oriented here. So this would be the small ribosomal subunit. And the important part of the small ribosomal subunit is this messenger RNA binding site. Okay, it's where the messenger RNA binds. The next part of the ribosome is this bit here, the large ribosomal subunit. And there are several different sites within the large ribosomal subunit that I want you to be aware of. So one is this A site. All right, now you can just remember it as the A site. It stands for the aminyl acyl tRNA binding site. And then we've got our P site. You can remember it as the P site, but it stands for the peptidyl tRNA binding site. And the E site is easy. The E just stands for exit. All right, so we've got our E, our P, and our A binding sites. The last thing I want you to notice on this ribosome is this exit tunnel right above the P site. The exit tunnel is where the polypeptide that's produced leaves the ribosome. Alright, so I'll show you the relevance of these EPA sites in a moment, but they are where transfer RNAs locate within the molecule. Amino acids get joined together, they form a big long chain, and that leaves the exit tunnel. Okay, so what I want you to do now is to draw a ribosome and label all of the parts. That will help you remember it. So we've got our small ribosomal subunit. And if you remember, it's the 40S subunit. And then we've got our large ribosomal subunit. There's our large one, and that's the 60S subunit. We've got our messenger RNA binding site there. I'm just going to write mRNA bind. Okay. And we've got our 3 sites within the large subunit. And they are our E, P, and A sites. E site is the exit site, and these are positions where transfer RNAs would locate. And then of course we've got our exit tunnel just above the P site, which is where our growing polypeptide chain, as it gets assembled, would exit the ribosome. Alright, so that's the lay of the land. That's what I want you to know about the ribosome. Okay, so let's see if we can tie all of this together. We've got 3 different kinds of RNA. One kind is our messenger RNA. Now, messenger RNA, you can think about that as our code carrier. Okay? The genetic code information that's in DNA is essentially transcribed and converted into the code carried by messenger RNA. Alright, our second player then is tRNA. tRNA, its major job is to carry amino acids. It's our amino acid carrier. And then our third player is our ribosomal RNA. And our ribosomal RNA, its job is the site of protein synthesis. It's where the amino acids are linked together, one after the other, in great big long chains that could be hundreds of amino acids long. Now, the sequence of amino acids is determined by the DNA sequence, so therefore is determined by the messenger RNA sequence. So which amino acid this is, which one of the 20, is determined by the code in messenger RNA, which was determined by the DNA code. Okay, so next we're going to look at transcription. So now we can have a much closer look at the process of transcription, and that's where we take the information that's in DNA, in other words the sequence of nucleotides, and we convert that information into a strand of messenger RNA. So we take the information and we produce a molecule which carries that information. So we're going to look. Okay, so first I'm going to go over a verbal description of this process of transcription. Then we'll look at some diagrams to see how it works. Then we'll look at some animations, and we'll also draw the process so you've got a complete understanding. Alright, so here's an overview then. This process of transcription is the synthesis or production of a messenger RNA molecule using the information in DNA. And usually we take a gene, because genes are what we refer to as the coding part of DNA, and generally what code for proteins. So it's the information in a gene, which is a strand of DNA, and that information is transcribed to produce a strand of messenger RNA. So transcription then produces messenger RNA from the code in DNA. And it involves this very important enzyme. This enzyme is RNA polymerase, and it's the key enzyme involved in transcription. And it does two major tasks. The first thing RNA polymerase does is it separates the two DNA strands. And it does so about 1 to 20 bases at a time. And then it moves along the double-stranded molecule, the DNA, and it rejoins the DNA as it moves along on the back end. So first thing, it unzips the DNA molecule and can zip it back up. Second is as it moves along the DNA molecule, only one of the DNA strands that we refer to as the template strand. So it uses the template strand to assemble a strand of pre-messenger RNA at about 40 nucleotides per second. That's incredibly fast. And that pre-messenger RNA strand has a sequence that's complementary to the DNA template strand. Okay, so we're going to focus on the process in eukaryotes, not bacteria. And transcription occurs in 3 stages. The first stage we refer to as initiation, and that's where RNA polymerase attaches to the DNA strand at this promoter region, and it's characterized by something called a Tartar box, and that TATA stands for a thymine and an adenine, and a thymine and an adenine. So at that sequence, that's our Tartar box, and that's called the promoter region. And that occurs at the start of a gene. And when the RNA polymerase attaches to that promoter region, it moves along the DNA strand in a specific direction, and it begins to separate the 2 strands. So the next part is elongation. So only one strand of the double-stranded DNA molecule that we refer to as the template strand, that's used to transcribe pre-messenger RNA. And the RNA polymerase moves along the template strand of the gene, and it assembles a strand of pre-messenger RNA that's complementary to the DNA strand. And the third stage is this stage termination. So once a termination sequence on the DNA is reached, and usually that's at the end of the gene, RNA polymerase stops its elongation and it leaves the DNA molecule. Alright, so 3 stages. Initiation is where it joins, elongation is where it moves along the DNA and produces a strand of pre-messenger RNA, and termination where the process stops. So once the pre-messenger RNA transcript is produced, it's modified before it leaves the nucleus, and we'll look at that RNA processing step in a later concept. Alright, so this diagram represents or illustrates initiation then. So here we've got our double-stranded DNA molecule. There's the 5' end and there's the 3' end to the 2 strands. This would be our promoter region, and it would have the Tartar box there. And there's our RNA polymerase, which is joined or attached to the gene at the start of the gene to the promoter region. Then it unpries the 2 strands of DNA, breaking hydrogen bonds, and then it begins only on the template strand to produce an RNA transcript. That's our pre-messenger RNA. So elongation then, as you can see, the RNA polymerase has zipped along the DNA molecule in this direction, So it moves along the template strand from the 3' towards the 5' direction. So it zips along. As it zips along, it pries apart the two DNA strands, and then it begins to assemble this messenger RNA transcript, or the pre-messenger RNA molecule. And it keeps moving along the gene until termination, until it reaches a termination sequence, and that says, okay, stop here. And that point, RNA polymerase leaves the DNA molecule, and we have a completed strand of pre-messenger RNA that we'll refer to as the RNA transcript. Okay, so I want to have a look in a little bit more detail at this initiation part of transcription. So we've got our DNA molecule. It's double-stranded. There's one strand, there's the other, and you can see we've got our 5' end and our 3' end. So at the beginning of a gene, we have this promoter region. And the promoter region is usually characterized by what we call this TATA box. And it has this TATA sequence. So there's our TATA box. And that sequence seems to be a fairly common one at the start of many genes. And it's this region that we call our promoter region. Now this will actually be our start point. But this is what RNA polymerase recognizes. Okay? All right, so first we actually get some transcription factors attached. Don't even worry about what they are. They're a bunch of proteins. They attach and they enable our RNA polymerase 2 to attach, to recognize and attach. So the RNA polymerase recognizes and attaches, and at that start point, it unzips the two DNA strands by breaking the hydrogen bonds, and then it starts only on one of the DNA strands that we call the template strand. It begins to use the DNA sequence in that template strand to assemble a strand of pre-messenger RNA, which we call the RNA transcript. All right, now this is important. It moves along the template strand in the 3' to the 5' direction, and so it's going to assemble a strand of messenger RNA in the 5' to 3' direction. And you'll have to go back to chapter 16 to refresh yourself on the 3', 5' directions if that's something you can't remember right now. Okay, so here's just a little bit more detail of what's going on during elongation within the RNA polymerase molecule. So up here we've got our non-template strand, and that's simply ignored. Down here we've got our template strand, and that's the one that we're interested in. And if we have a look at the sequence in the template strand, back here we've got a T. Well, the complementary base to that is an A. Here we've got an A, and the complementary base in RNA is the U. Here we've got a G, so the complement to that is a C. So you can see that using the code in the DNA, the RNA polymerase assembles a complementary strand of messenger RNA, and it moves along the template strand quite quickly, 40 bases a second roughly, unzips, reads the DNA base, and then it will pull in an RNA nucleotide, join the nucleotides together to form a strand of messenger or pre-messenger RNA. So that's the way the process works.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
Okay, so I want to take as an example the human insulin gene. Now remember, a gene is just a length of DNA that codes for a protein. And in the case of the insulin gene, that gene codes for the insulin protein. Alright, so a gene is just a length of DNA, but it's a little more complicated than that. That gene is actually composed of several different regions. We have regions called exons and regions called introns. Okay, now in the case of the human insulin gene, there are 3 exons and 2 introns. Now, the exons contain coding regions of that gene. So exon 1 and exon 2 and exon 3 actually form the information part of that gene, which are used to code for the protein. Intron 1 and intron 2, you can think about those as being useless. They're irrelevant, they're not needed, and they actually need to be removed. So I'm going to show you how they get removed. Now the human insulin gene is actually 1,430 base pairs long, and that whole gene undergoes transcription to produce the pre-messenger RNA. And in the pre-messenger RNA, we have all 3 exons as well as both introns. And that pre-messenger RNA needs to be processed, and we'll look at that processing in the next section. So the messenger RNA or pre-messenger RNA that is produced by transcription, it's not ready to leave the nucleus and become functional. First, it needs to be modified. So we're going to look at that process of RNA. So here's our length of pre-messenger RNA, and it consists of an exon fragment, an intron, another exon, another intron, and another exon. Now this is specific to the human insulin gene, which I'm using as my example. Other genes may have different numbers of exons and introns, and they're going to vary in length as well. But that pre-messenger RNA undergoes messenger RNA modification, and we get a length of functional messenger RNA, which is then ready to leave the nucleus and be translated on a ribosome. So we're going to look at this process of messenger RNA modification, how we go from this to fully functional messenger RNA. Alright, so the pre-messenger RNA is modified after transcription, and before it leaves the nucleus to be translated by this process called RNA processing. The first part of the RNA processing is to add what's known as a 5' cap to the 5' end of the messenger RNA. The next thing that's done is a long chain of A's, a long chain of adenines is added to the 3' end of the pre-messenger RNA, and we call that a poly A tail. So we add a 5' cap to the 5' end and a long chain of A's to the 3' end, and that's the poly A tail. So these modifications help the messenger RNA move out of the nucleus. It helps protect it from degradation by nuclease enzymes, which are present in the cytosol, and they're there to break down any nucleic acid. And so they might break down the messenger RNA if it wasn't protected by, in this case, the poly A tail. And they also help it to attach to the ribosomes. Alright, so RNA processing then. The first part of RNA processing is we take our pre-messenger RNA and we add a 5' cap on the 5' end, and we add a poly A tail on the 3' end. That's quite simple. So this diagram's out of your book, and you can see that there's the 5' cap and there's the poly A tail. I'm not going to worry about that, but it's this region here which consists of some introns and some exons, and we've got a start codon there and a stop codon there. So most of this is sort of the business region. Some of it will be removed and some of it will be used to code for a protein. Okay, so the next part of modification of the messenger RNA is called RNA splicing. Remember, most eukaryotic genes have got these non-coding sections of DNA in them called introns. They don't do anything, they're non-coding. And you've got coding sections of DNA that we call exons. These are the important ones. These are the ones that are exported from the nucleus, and they're the ones that have the information that code for proteins. So both the introns and exons are transcribed into the pre-messenger RNA, but the intron sections have to be removed by the process of RNA splicing. So it's quite simple. Once spliced, the messenger RNA can leave the nucleus and go join a ribosome. Alright, so there's our slightly modified pre-messenger RNA. It's got the 5' cap and it's got the poly A tail, but it still has introns, and we're going to get rid of those introns. So we undergo messenger RNA splicing. Enzymes come in and they make a cut there and a cut there, and another cut there and another cut there. The introns are then removed. They're gone. So then we have this situation, and the different pieces of messenger RNA need to be joined back together. And so there are other enzymes that join these together. And then we get something that looks like this. We've got our 5' cap, our 3 exons, which are the coding region, and our poly A tail. This now is ready to form messenger RNA. It can leave the nucleus and go out into the cytosol and find a ribosome. Alright, and here's the diagram from your books showing essentially the same thing. Here's our 5' cap. Here's our exon, which in this case is, it says from 1 to 30 base pairs long. There's our intron, we're going to get rid of it. There's our other exon. There's our other intron, we're going to get rid of it. There's our other exon, and there's our poly A tail. So once we get rid of the introns, here we have the exons all joined together, and if we add up their length, we're going to get something around there. Now, this example doesn't match with the human insulin gene example I've used, because these numbers, these lengths are quite different, but it just gives you an example. Okay, good, good. Okay, so now we're going to look at what happens to the messenger RNA after it leaves the nucleus and how it gets translated so that the code in messenger RNA can be used to make a protein. So that's the process of translation. It's taking the information in the messenger RNA, translating it, and producing an actual flesh and blood protein. All right. Now, proteins don't look like green boxes, but this protein here is the human insulin protein. Now, proteins, remember, are either polypeptides that are folded or are made of several different polypeptide chains. Now, I know it's kind of small, but this shows a polypeptide chain of the human insulin protein. Each one of these little circles is a different amino acid. So you can see that they're amino acids linked in a big long chain. Now again, that just shows the linear arrangement of the amino acids. This diagram here shows you the three-dimensional shape of the human insulin protein. And you can see that there are helices and there are some sheets, and it's actually got some quite nice symmetry. But this is what it looks like as a chain, and this is what the actual molecule looks like after the polypeptide has become folded. Alright, so we're going to look at this process of translation, taking the information in messenger RNA and using it to direct the sequence of amino acids in a polypeptide chain. Okay, so let's have a closer look at this process of translation, and we'll go through it step by step. So the first thing then is the messenger joins to a small ribosomal subunit at its 5' end. So I'm going to draw a small ribosomal subunit. And remember, it has that little attachment point for the messenger RNA. And now I'm going to draw my messenger RNA molecule. And I'm just going to make up a sequence. We'll get a U and an A and a G and a C and a G and a C, C, A, U, U, A, and so on. I won't go ahead and draw it all. Alright, so the next thing then is a large ribosomal subunit joins, and that makes a functional ribosome. There's our large ribosomal subunit. Now if you remember, the large ribosomal subunit has those three sites. There's the E, the P, and the A site. E, P, A. Alright, and there's an exit tunnel there. Okay, now I've not properly matched up codons on the messenger RNA to these locations. Alright, to the E, P, and A sites. But we'll look at that in a better diagram next. I just wanted you to get the picture that the messenger RNA joins to the small ribosomal subunit, and then a large ribosomal subunit comes and joins those. Okay, so the next thing then is that messenger RNA moves through the ribosome one codon at a time in the 5' to 3' direction. Transfer RNAs then carry amino acids that match their anticodons to the codons in the messenger RNA. And that means that you're going to get the correct amino acid sequence in the polypeptide, which is dictated by the sequence in the messenger RNA that was dictated by the sequence in the gene. So remember, the ribosomes have got three sites, the E, P, and A site. The amino acids are joined together to form a polypeptide, and the growing chain leaves the ribosome via the exit tunnel. Okay, so let's take a closer look at translation, and we're going to start this process by looking at the start of translation. So here we've got our small subunit. Here we've got our messenger RNA molecule, and it's attached to the small ribosomal subunit, at the messenger RNA binding site, and that's at the 5' end. And there's our 3' end of the messenger RNA. So, as soon as the messenger RNA binds to the small ribosomal subunit, we get an initiator transfer RNA come flying in. And usually that initiator transfer RNA has this amino acid attached to it. MET stands for methionine. It's the amino acid methionine. And it binds there because at this location on the messenger RNA is a start codon. Now the start codon has that sequence there of AUG. And the initiator transfer RNA has a complementary anticodon, UAC. So there's a codon-anticodon match just there. Now when the initiator transfer RNA joins, then we get our large ribosomal subunit come flying in, and the large and the small ribosomal subunit are joined together. As soon as that happens, we've got a functional ribosome, and we process through translation very, very quickly. Okay, so now I'm going to draw your attention to some of these sites. Remember our E site, our P site, and our A site. Alright, so now we're going to look at the next. Alright, so the next thing that would happen then is we have a transfer RNA at the P site. The A site is vacant, and a transfer RNA comes flying in to the A site, and it will match anticodon to codon. So now we have transfer RNAs at both the P site and the A site. Once we've got these two amino acids next to each other, a covalent bond is formed between these two amino acids. So now the transfer RNA that was at the P site has lost its amino acid because it's been joined covalently to the amino acid that belonged to the transfer RNA at the A site. So by joining together amino acids, you can see how we get a growing polypeptide chain. So the messenger RNA is going to move along one codon, and as it does, this transfer RNA suddenly hits the exit site and it's detached and kicked out. The transfer RNA that was at this A site is now at the P site, and that leaves the A site vacant, and we start the whole process again. Another transfer RNA will come flying in. They'll join their amino acids together. It will bump down one codon, and a transfer RNA will exit without its amino acid. Alright, so hopefully you can see how that growing polypeptide chain, each one of these shapes is a different amino acid, grows. As the messenger RNA moves through the ribosome, other transfer RNAs come in, more peptide bonds are formed between amino acids.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into three stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anticodon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated. Three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
Click on each of the animations. They are both absolutely brilliant animations showing the process of translation. Alright, so once the polypeptide chain begins to leave the ribosome, it starts to fold into a functional protein, or something that will approach a functional protein. Now remember, the primary structure of a polypeptide is simply the sequence of amino acids in the chain. And we already have a long polypeptide chain made of a chain of amino acids. So then it begins to fold into its secondary structure, and that is composed of alpha helices and those beta pleated sheets. You might have to cast your mind back to chapter 5 when we talked about macromolecules to remember that. They're both held together by hydrogen bonding. Once it forms its secondary shape, it will fold into its tertiary shape. And if there are additional polypeptide chains, it will form a quaternary structure. Now, that may yield a functional protein, although the polypeptide chains are very often modified, and they can go through extensive modification in the Golgi apparatus, for example. So if you have more than two polypeptide chains, they can join together and they can form a functional protein with quaternary structure, as we mentioned. So those proteins can be used within the cell. They might be something that's important within the cell, or they can be packaged into vesicles for export from the cell. In the case of the human insulin protein, it's modified and then it's packaged into vesicles, and it's exported from cells of the pancreas where it's produced, released into the bloodstream, and there it can help regulate blood glucose levels. Pretty cool stuff. Okay, so I want to summarize everything with a few diagrams. Okay, so this has a little bit more than I want you to know, but I do want you to know transcription. We've talked about that before. That's where we take the code in DNA and produce a strand of pre-messenger RNA. Here we call it the RNA transcript. The RNA transcript then goes through RNA processing and it produces a functional strand of messenger RNA that leaves the nucleus. Don't worry about that, and don't worry about that, and don't worry about that. The amino acid activation, don't worry about it. What I want you to know is that there are transfer RNA molecules with amino acids attached to them. Okay, so messenger RNA now comes and joins to a small subunit, and then the large subunit joins, and then we get this big translation complex. We have our E, P, and A sites. The messenger RNA bumps through the ribosome in that direction. There's our 5' end and our 3' end, so it bumps through in the direction 5' leading, 3' lagging, and it bumps down one codon at a time. And here we have a codon, there's another codon, and there's another codon, and the transfer RNAs match anticodon to codon, anticodon to codon. And as they come in and join, their amino acids are joined together to form this growing polypeptide chain. That summarizes just all of it. Again, don't worry about that. All right, so now you can see everywhere from transcription, the RNA processing, and translation. All right, there's nothing like watching it as a movie, so there's a movie I want you to watch next, and it kind of summarizes a large amount of the process. Okay, so you understand the process of transcription, RNA processing, and translation. But now we've got to crack the code. And what I mean by that is, let's take the DNA sequence and see if we can use that to determine the amino acid sequence in a protein. All right, so let's take a closer look at cracking the code. We're going to see how we can go from a DNA sequence to a sequence of amino acids in a protein. So the first thing that we know is that messenger RNA has codons, and each codon is 3 bases in a sequence. So I'm going to draw a couple of messenger RNA codons. I'm going to draw a U, a G, and a U. So there's one codon, and then I'm going to draw a C, a G, and a U codon, and then I'm going to do a C, and a U codon. So these are our codons in our messenger RNA. Now, of course, this messenger RNA molecule could be much, 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 much longer. All right. Now, each codon has a corresponding anticodon on a transfer RNA. So now I'm going to draw the corresponding anticodons. That would be an A, a C, and an A. And there I'm going to draw a transfer RNA. I'm going to kind of represent it like this. And so our corresponding anticodon to this second codon would be G, C, and A. There's our corresponding anticodon on another transfer RNA. And then we've got this last codon of CCU, and that would be a G, G, A. All right, there we go. So these then are our anticodons. Now what we also know is that at each transfer RNA has an amino acid that's specific to its anticodon and therefore specific to the codon on DNA. Now we know from a database that the anticodon of ACA has the amino acid cysteine, which we abbreviate to CYS. And we know that the anticodon GCA has the amino acid arginine that we abbreviate to ARG. And then we know that the anticodon GGA has the amino acid proline that we abbreviate to PRO. So now we've got our amino acids. All right, so we've got our amino acids, we've got our anticodons, and we've got our codons. All right. So what we're going to do is we're going to cut out the middleman. We're going to ignore transfer RNA. We know the amino acid for each codon in the messenger RNA. In our previous example, we knew that the codon UGU corresponded to the amino acid cysteine. And we knew that the codon CGU corresponded to the amino acid arginine. And we knew that the codon CCU corresponded to the amino acid proline. All right, so all we need then are the codons. The messenger RNA codons enable us to determine the amino acid sequence. Okay, so I want you to remember that there are 20 different amino acids, and there are 4 nitrogen bases. So with 4 nitrogen bases, that means we've got a total of 64 possible codons, because it ends up being 4 nitrogen bases to the power of 3, because there are 3 bases in a codon. So if we have 4 times 4 times 4, that gives us 64 total possible combinations. So that gives us 64 codons. Now, because we've only got 20 amino acids very often have more than one codon that code for them. All right, so we're going to use this table to help us figure out which amino acid belongs to which codon. So we'll work through a couple of examples. Let's take we've already been used to looking at, the U, G, U. Now this is our 5' end, and this is our 3' end. All right, so this one here represents our first base of the codon, and this represents the second base of the codon, and this one represents the third base of the codon. All right, so now let's use this table. Over here we've got our first messenger RNA base at the 5' end. And our first messenger RNA base was uracil. So we're going to start just there. All right, our second RNA base, just here, second RNA base, is G. So we take that G just there. So now we know we're in this box here. Okay? Because we had U here and we had G here. Now let's take our third nitrogen base of U. There's our third nitrogen base, and we've got to make a choice of these ones here. So we've got U. So now we can see UGU, and that corresponds to the amino acid cysteine. All right, let's look at another example. Our other amino acid that we were looking at was a CG U. Alright, so let's go find our first C. Our first C, and I'm going to change pen colors. So our first C would be over here. Our second one is G, and that would be over here. And so that puts us in this box here. Okay. And our last one is U, which would put us just here. So there you go. CGU, and that gives us arginine. Alright, we'll do our last example then, and I'll choose a different color. Let's go with that nice teal. So we're going to take the the codon CCU. Alright, here's our first one, C. Here's our second one, C. That puts us in this box here. Our third one is U. That puts us here. And there we've got CCU, and that gives us the amino acid proline. Alright, so hopefully that's enough practice to see how you use this table. Okay, so we're going to do a practice problem now. Alright, we're going to take a DNA sequence first. And here's our DNA sequence. Alright, so the first thing we're going to do is figure out what our messenger RNA sequence would be from that DNA sequence. Alright, so I'm going to help you here. This would be our 5' end. And so A would give us a U. Okay, the T would give us an A, the G a C, G, G, C, A, U, U, A, C, C, and there's our 3' end. Alright, so that's our messenger RNA sequence, and you can see that we can break this up into codons. Alright, there's some of our different codons. Alright, so the next thing we've got to figure out is the amino acid sequence. So I want you to go get the table in your textbook, and you're going to read off of that table in the same way that I just went through those examples, or you can pull up one of these tables on the internet. But so we're going to look at U, A, C and read that off the table. So U, A and C would give us the amino acid tyrosine, TYR. There. Now let's have a look at our next one, GGC. Alright, GGC would give us the amino acid glycine, LY, as we would read off of our table. So then we're going to take the next codon, AUU, and we'll read off of our table to see what that gives us. AU gives us the amino acid, ILE. I think that's, I can't remember what amino acid that one is, but I'm going to write the abbreviation, ILE. I think that might be isoleucine, but I'm not 100% sure. Alright, let's take our last one, ACC. ACC, I'm reading off the table, and that gives us the amino acid THR, which is threonine. So there's our amino acid sequence. So we can go from DNA sequence to messenger RNA sequence to amino acid sequence in the protein. Alright, very cool. So now let's do practice problem number 2. Alright, and I want you to be good at these practice problems because in an exam I may give you that table and I may ask you to figure out protein sequences from either DNA sequences or messenger RNA sequences. Alright, so here's a DNA sequence. Alright, now I want you to be careful with this next practice problem. We've got to get our messenger RNA sequence. Now, if this is our 5' end on the left, this must be our 3' end of the amino acid of the messenger RNA. So then we're going to have an A, and a G, a C, a G, and a U, an A, G, and a U, and then a G, a C, and there's our 5' end. Alright, now we've got to get our amino acid sequence. Here's what we cannot do, so don't make this mistake of simply reading AAG in that direction and think that this is our first codon. We've got to start down here at this 5' end, and we've got to say this is our first codon, and we've got to read it in that direction. And then we've got to say this is our second codon, and we've got to read it in that direction. And then we say this is our third codon, and we read it in that direction. And this would be our fourth codon read in that direction. Because the table reads the codons in the 5' to the 3' direction. Alright, so now we've got a CCG codon. CCG gives us the amino acid proline. Now we've got to get a UGA. Alright, UGA. Oh, I can't believe it. That is actually a stop codon. Alright, now if that was a real messenger RNA sequence being read by a ribosome, we'd stop. We'd stop right just here and we would not proceed. But I am going to proceed just because I want you to be able to do the example. So that was just fluke that ended up being a stop codon just by coincidence. Alright, so now we've got to look for a UGU. Alright, UGU. And that gives us cysteine, CYS. Now let's look for a GAA. GAA, and that gives us ASP. I think that's aspartic acid, ASP amino acid. Alright, so that's how we would do that problem. You've got to make sure you get your directionality correct, but hopefully these two practice problems have helped you. If you need more help, come talk to me in office hours, but in both your chapter exam and final exam, I will give you problems like this, and I'll always tell you the directionality, and I'll give you some scratch paper and pencil so you can write down exactly what we did on the screen. Alright, so good job.